episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor. Welcome, everybody, again to another episode of our show. Today, we're bringing you another really fascinating guest who is focused on creating a better tomorrow for all of us. Today, we have the honor of being joined by Dr. Harold Katcher, who is currently the Chief Scientific Officer of a company called Yuvan Research. It's a biotech company exploring the development of novel young plasma fractions focused on rejuvenation treatments in mammals. Most recently, Dr. Katcher was the Academic Director for Natural Sciences, Asian Division of the University of Maryland Global Campus, and throughout his career, Dr. Katcher has been a pioneer in the fields of cancer research, including the development of certain aspects of gene hunting and sequencing. He was one of the discoverers of the breast cancer gene BRCA1, as part of Myriad Genetics, and he has broad expertise in the areas of bioinformatics, chronobiology, biotech. Dr. Katcher has literally thousands of citations across the scientific literature, with publications ranging from protein structure, biotech, bacteriology, bioinformatics, and biochemistry. And his new book, entitled The Illusion of Knowledge, will be out on September 4th of this year. A lot to talk about. Dr. Harold Katcher, thanks so much for joining us on the show today.
You're very welcome. You make me sound like jack of all trades, master of none. That's been my life, and I've always been looking at the bigger picture.
Absolutely. And Howard, I'd like to start off there, with the bigger picture, and sort of give you the floor for a couple of minutes. Obviously, you've been involved in a lot of areas over your career. Talk for a couple of minutes about how you got interested in aging, longevity, and if you could spend a little time. Obviously, we're in this space where everyone has their theories of aging that they like to promote. Talk a little bit about your current theory of aging, and we'll get into plasma and everything else later on, but walk us through it for a little bit, if you would.
For me, aging is simply a continuation of development. I mean, we name stages in development like zygote and blastula and gastrula and that kind of stuff. We even go further. We go up from embryo to fetus, from fetus to newborn, from newborn to toddler, from toddler to preschool, from young school, etc. We recognize stages as development. But according to all current aging theory, other than mine, and others like Steve Horvath, we stop at adulthood. We stop development, and we just start deteriorating. And the deterioration is random. Well, I say that's wrong, and that's contrary to anybody's impression of what life is about, because we see life goes through various stages. We can recognize an older adult, whether he's middle-aged or old-aged or etc. So clearly stages are there. Most recent scientific evidence, actually a metabolic study from Science last week, showed that you can divide the post-adult lifespan into at least two other, middle age and old age and young adult. So at least three stages, and I agree with all of them. And you can probably subdivide them. And the stages are the same in all mammals, in all placental mammals. So that's why Steve Horvath developed a clock where you can put in DNA of any placental mammal, a mouse or if for some reason a shoe came to mind, it's not placental, a mouse or a lemur or whatever, and it'll give you an age that'll depend on what species it is, but all from the same pieces of DNA, all from the same homologous DNA sites. So for me, aging is just a progression of life stages. People don't want to confront that because it means that death is inevitable.
Yeah.
However, we also discovered something, sometimes once you confront your fears, you can actually solve your problems. And we found that aging, in fact, was controlled systemically, controlled by factors in the blood. Actually, we didn't find that. People found that for decades.
Yep.
Nobody talked about it because it didn't fit into any current aging theories. So instead of changing aging theory to fit the facts, you just ignore the facts.
And unfortunately, we see that a little too often in lots of areas of the biosciences. But keeping that in mind, and thank you for introducing the concept of continued development, theory of aging, and now you brought up blood, and I'd like to transition in first for some of the folks listening that may not be in this space or might not have a life sciences background. Obviously, we have several quarts of this material called blood flowing through us, and when we get rid of the blood cells, we're left with this stuff called plasma, which makes up, it's comprised of a certain amount of
about half.
Okay, about half, and it's chock full of stuff. I'll just call stuff right now, and we'll get into things later on of what's there. But I saw a paper a couple of years ago where I think they identified something like 4,000 different factors and proteins and microRNA. It's just made up of it's a wonderful cocktail of things, and we'll call it things for now.
Absolutely.
Introduce us to plasma. How do you think about plasma as a general component of what keeps us, keeps me here and not bleeding to death as we're talking, and at the same time, what else? Walk us through that.
There's a lot of functions. It's antibacterial, for example. It has the complement system that attacks bacteria. It has the antibodies. It has the growth factors. It has the hormones. It has many, many factors. And I cannot, without violating my IP, specify any particular factors.
Sure.
So all I'll say is that people have already discovered factors that both promote youth and inhibit youth. For instance, there are people who are interested in a growth factor, GDF11. There are people who are interested in oxytocin. People are interested in various things.
Right.
Let me just say one thing now. From a genetic point of view, if there were a single item that caused aging or youth, then a mutation in that single item would result in all kinds of aging mutations. We'd see immortals walking the streets. We'd see people who lived for an hour and a half and died of old age. We don't see that.
Right.
So we know that can't be the case.
Let's go further with that. Now, understanding we're not going to get into any IP, proprietary information, but I do want to starting then from plasma. And you rightly point out lots of things there. There's not going to be a single magic bullet floating around. You, from what I understand, work with what we'll call some people call them cocktails, some fractions, depending on where we are. But at the end of the day, the concept of a combination of stuff, mostly say that's the good
Correct.
stuff, we get rid of the bad. And this is it's a fascinating area because I think I spent the last 30 some odd years in the pharma industry, and I think there's a lot of people that are unaware that this in terms of combinations, natural combinations, were quite prevalent in the pharmaceutical industry in the first part of the 20th century. I give the quiz that one of the most popular drugs ever prescribed, Pfizer's Premarin, is a mixture of estrogens derived from horse urine. It has no real composition. It's a cocktail of stuff. Talk a little bit about, obviously not what your fraction is, but in studying the fraction, how you decided to sort of whittle down the thousands to whatever you have in your cocktail. What type of tools do you use? You look at proteins.
I tell you what, all the ideas that I got about the fraction, I got from the literature.
Okay. Fair enough.
It was only a question of connecting the dots. I can't tell you which dots I connected.
Not a problem.
But it's all there. You just have to look through the literature, you'll come up with the same thing. I just happen to be lucky. Well, if you read my book, you'd find that perhaps it was more than luck.
Gotcha. Not a problem.
But for me, this was the ultimate question always in biology, life and death. Nothing else really mattered to me. So, yes, I studied protein structure just because it's interesting. Now artificial intelligences could do it much better than I could ever do it. But I tried to look at the big picture of what was happening and how everything was coordinated. And I try to figure out how, the concept that most people have of death, of life and death, is a picture of a car, an automobile. Gradually it rusts, it wears, the tires wear away, you replace them, but after a while it be no longer cost effective. And turns into a jalopy and it heads for the junk shop. But that's not how living systems work.
Right.
That's a concept of entropy.
Right.
Concept of things fall apart. Mountains wear down into valleys, valleys fill up into plains. This is a natural progression of things, and that is a natural progression of things under certain circumstances, but not the circumstances of life.
Right.
That's life is what you call an open system. Any living thing.
Yep.
We shouldn't even speak of life. It's a process we don't understand whatsoever.
That's true.
And any living thing violates the principle of entropy by growing more complex with some part during part of development, at least. And we're an open system. We take in energy and we take in matter.
Right.
And entropy only applies to closed systems that don't take in energy and matter.
Right.
So, I don't want to bore you.
No. Yeah, you're spot on. But I mean, we come from a similar direction in our thinking here. Well, let's move forward. Then you went through the literature. You developed this proprietary fraction. Come May 2020, you publish Reversing Age, Dual Species Measurement of Epigenetic Age with a Single Clock. Steve Horvath, who you mentioned at the beginning of epigenetic clock fame, is one of your co-authors. Talk about May 2020. Talk about this work and what you are excited about. Take us a little bit through it, if you would.
Well, you know, it's a long story, and I don't want to bore you, but my original idea occurred in 2013, where I developed what I called heterochronic plasma exchange.
Right.
The idea was that if there were pro-youth factors and pro-aging factors, then the simplest way to do things is to simply replace the plasma of an old person with the plasma of a young person.
Mm-hmm.
And perhaps if you did it enough times, it would stick, as it were. That is, the person would retain the youthful phenotype. So Akshay Sanghvi, who is a financier and entrepreneur, and a devotee of biological science himself, invited me. This was actually the second time. The first time I was taken in by some people and agreed with a plan of theirs that never worked out. But the second time, I had to go to Mumbai in order to do research. Mm. Now, Mumbai is probably not the center of the scientific world. And the university I was there, NMIS, seemed to me to be a very different kind of place from anything I've seen in the United States, because they had a lot of equipment, mm-hmm. but it was all common equipment, and nobody used it. Some of it's been left lying there because basically it was for student use. The dean, Dean Bob, who is an exceptional person and an exceptional dean, said that the Indian government, when they give grants to researchers, make no allowance for the schools, which means that there's absolutely no motivation for the schools to hire researchers, because they pay them for nothing. They make their money through teaching students. So it's just, what seems like wise policy changes can really turn in the other direction. Yeah. So, any case, it took there's a lot of bureaucracy in India, and it took a long time to get order stuff, but we could do things there that we cannot do here, because we just lack, because there's COVID crisis. Mm-hmm. We lack the manpower. Equipment is delivered late, and we have to get it from exotic places. Yeah, it was easier to do research in Mumbai, and I had a bunch of graduate students who actually knew what they were doing. Yeah. So we try to try total plasma replacement with rats, heterochronic mm-hmm. plasma exchange.
Okay.
We couldn't. We had some German researchers said they had a machine to do such a thing, and they showed pictures of it, and we could never somehow never make contact with the suppliers of the machine. We could never get the correct details. We could, and we tried it manually then. Mm-hmm. To manually extract blood from a rat, centrifuge it, replace the plasma with young plasma, similarly obtained. But rat veins are too fragile to allow that to happen, so we couldn't do it. Mm-hmm. And I was stuck. And then I said, well, okay, because you were only allowed a certain number of visas to India per year, a certain number of visits. Now I have a 5-year visa, but that's immaterial. So I came up, I said, well, let's isolate those factors that are pro-youth or that determine aging, and let's inject it in large amounts into animals.
Mm-hmm.
And I left for the United States, and I was relatively sure that it wasn't going to work. I didn't expect it to work whatsoever. Well, I mean, obviously expected it to work to some degree, or I wouldn't have come up with it. And then I got the email that it did work. The increase in grip strength, a decrease in inflammatory cytokines, and we did about 30 other tests, and they all agreed. They all seemed to mark about the same life stage. You know, your HDL decreases as your age, your LDL increases, your low density lipoproteins, your bad cholesterol increases as you age. But we found this is just part of the aging phenotype. This is not the result of lifestyle or some fault on your part.
Right.
This is what happens, and it can be reversed. And that's what we showed. Every single one of them can be returned to youthful adult levels. So I wasn't convinced. Well, of course I was, but I wrote to Steve Horvath, who I'd read about in Nature. Mm-hmm. They had a 2-page spread on him.
Sure.
It's not something that Nature often does. And I said, could we send you, could we make a rat clock? So this was my idea. And it turns out that Steve had similar ideas. Mm-hmm. And he was collaborating with Rodolfo Goya down in Argentina.
Yep.
And together we made a rat clock, a Sprague-Dawley rat clock. So then we could send Steve samples, and he can evaluate whether or not, in fact, there was a reversal of I didn't expect it, quite honestly. I figured out any number of mechanisms whereby the epigenetics was somehow bypassed or whatever. But when Steve came back and said it works in an email, and then the result showed something like a 54% reduction in age, which he had never seen anything like that. None of us, pardon me, nobody had. And then I said to him, Steve, I want you to check this so-called young DNA. Is there any way of telling whether really somebody might have just slipped young DNA in? And he came back to me in a couple of days and said, no, this is definitely not young DNA. Mm-hmm. Even though his age markers said it was young DNA. So that's basically the story. Almost now we have a continuation of the story.
Right.
A man named Didier Cornell, Belgium, gave us $25,000 to do a longevity study.
Okay.
This was not our immediate interest, because our immediate interest was getting it on the market. To get it on the market, you have to go through the FDA, and to get through the FDA, you have to show that it cures a disease, or
Sure.
pardon me, or at least ameliorates a disease.
Right.
And aging is not considered a disease, which is everybody's problem in the business, as it were.
Sure.
So what do I want to say? So Didier financing us allowed us to do this study without any motive other than to find out whether or not, in fact, reversing the age of the mice extended their lifespan. And that study is in progress, but I have a recent video.
What's going on in this video?
In this video, we simply see two rats, both the same age, one treated, one untreated. The one has a big C on its back, meaning control.
Mhm.
One has a big T on its back, based on pieces of paper that are pasted to their backs. They don't seem to mind. I find that odd, but you can see that the rat that is the control rat, the old rat, the other old rat,
Mm-hmm.
just kind of is a blob. It's just kind of as a blob, just sits there, just has no definition really between its head and its shoulders. It's just one mass, and and it just sits there. And the other rat is sleek and it has definition. It is constantly looking around. It's jumping up. It's climbing to the top of the box, the container it's in, and looking out over and obviously much more active and energetic. And that's about 90 days after the second treatment. We find the first treatment lowers the epigenetic. We don't know about the epigenetic age. We have yet to do a longitudinal study together with Steve Horvath. But we know that the first treatment apparently lowers their age somewhat. The second, but the age starts climbing up more rapidly than normal.
Mm-hmm. Mm-hmm.
The second treatment brings the age back down further. It always brings it down to maximum young adult. So
Mm-hmm.
it's not like Benjamin Button, turn into a baby and,
Right. Sure.
and just disappear. But it is very much like the Benjamin Button effect, and I'll tell you why. But what was I saying?
You're talking about the sequence of treatments and the respective amount of anti-aging each time.
The second treatment, we don't see any increase in aging. It doesn't even seem to be the normal increase in aging that we see with the controls. I imagine it's just a question of time, but just before we do a one shot every 30 days. 30 days in a rat's life is about 30 days is 2.5 years. We do it every 90 days, my mistake. So it would be equivalent to every 8 years.
Got it.
Yeah, my mistake. It would be equivalent to every 8 years in a rat's life. It gets a new treatment. So after the second treatment, all the signs were that it was young again. And now at the end of the second treatment, and this is after another 8 years, rat years, we give it a third treatment. But before that third treatment, this video that I talked about was taken. So by the end of the second 8 years, they had gone back to apparently looking youthful, behaving youthfully, as well as having all of the blood signs and biochemical signs and physiological signs. Nobody's interested in that. People are interested in being young again.
it's interesting, when you talk about it and the way you think through it, because obviously, as you said, it's not one and done. But when you think, okay, we're putting this we have a circulatory system, it's going everywhere.
What do you mean by one and done?
I mean, well, maybe I'm wrong, and maybe you believe it, but
No, you tell me.
do you believe 10 years from now, if I'm your patient, I'm one shot and then I don't come back for 8 years, or
It's a series of 4 shots,
Okay.
but it takes a week. But yeah, and then you don't come back for 8 years. Where's the profit in it for me? I'm not in it for the profit, but
No, it's interesting though, because once again, it's just as you going back to sort of the theory of aging and the way we've erroneously thought of things in terms of the car model, and that there is this body. I'm 53 now. I'm pumping out a bunch of 53-year-old stuff that's going into my old plasma now, and it's changing. And it's interesting the way you think about, okay, I'm putting the first shot in, something's happening with the epigenetics, and then I get the second. It's just interesting to me, because we don't hear it as many people think about sort of the way, but life naturally flows. As I said, I'm not the car. I'm not rusting one day. This stuff is happening over time, and it's just interesting the way you think through it. Obviously, we don't know what whether, as you said, it might be 4 shots, it might be 6. Who knows? Maybe it's 2. But it's interesting that you're giving it these thoughts now.
Could be 1, actually.
Who knows? Yeah, I mean, it's interesting. Do you envision? Because I know, as you mentioned at the beginning, there's other people that are playing around with the plasmapheresis and thinking about, okay, let's just take bad stuff out. Do you ever see the need to combine it with we pump in the good, but we want to also pump out some bad stuff at the same time?
That's what I wrote in 2013.
Okay.
That's what a chronic plasma exchange was.
Right.
Pump out all of the bad plasma, old plasma, and pump in all of the new plasma. Mm-hmm. So at the same time, you get rid of the aging factors, get the pro-youthful factors. This other group that you talk about, the convoys, and they, for me, are what led to everything. Mm-hmm. But unfortunately, their recent experiment, unfortunately for them, their recent experiment was kind of spoiled by the news that actually Dobry Kiprov was right, and young albumin actually is a pro-youth factor.
Right.
It's not the pro-youth factor.
Sure.
It's a pro-youth factor.
Right.
So I proposed doing it in 2013. We actually had, I think I can't discuss it. I'm sure I can't discuss the name of the proposed patient, but a billionaire who was willing to try this.
Mm-hmm.
And I had the cooperation of several people who are now competitors. But at the last moment, he chickened out. His doctor said, "No, I don't think this would be safe." And next year, he died of old age.
Mm. Those damn billionaires! I was dying.
Poor guy. He could. What would have happened? The worst that could have happened, he would have lived a year less.
Harold,
I suppose I don't blame him.
Yeah, what are you gonna do? Talk about the book for a couple of minutes. Why? Tell us. It's coming out September 4th. When did you decide to write it? What can we expect from it? And
Well, okay. COVID, of course,
sure.
Had me stuck in the house with nothing to do. And I thought writing a book might be a good idea. I just wanted to write down my personal history because I thought that this perhaps would be historic. Mm-hmm. Certainly, if we can apply it to human beings, we will. It's a proof of principles there. Now it's a question of engineering, and that's what I'm working on now. It's we've made breakthroughs and inventions galore.
Mhm.
So we're trying to upscale everything
Right.
to rats are a very poor market. And they don't really care if they live longer. They unfortunately, we're the only animals that know that we're going to age and die. It seems a little bit unfair, but maybe now our intelligence has brought that fairness back.
Got it.
So, what did you want me to say? Yeah, I don't understand why nobody has done heterochronic plasma exchange. I asked Dubry, he was originally going to do it, to this guy. Dubry, that's how I met Dubry Kiprov.
Sure.
He was a Los Angeles physician whose specialty was plasma exchange.
Right.
And he was contacted and he was willing to do it. He wanted off-the-shelf plasma because he believed that correct, that young albumin who was the secret. It's not. But so we had arguments. Ultimately, it didn't matter because our financier, our potential first patient, got scared and the whole thing was off, and that's that.
Mhm.
They went on to form their own company.
Yep.
And I was left. None of them followed my advice. I don't know why they. All right. Anyway, I was left, and Akshay contacted me, and I went to Mumbai, and the rest is history.
Gotcha. One quick one. You just stimulated something in my brain for a moment there when you were mentioning the rats not caring. But I've done a couple episodes recently, different groups that are working in dogs. Have you had any pet owners that come to you that want to try out?
Yes.
Okay.
Yes, and that's definitely one. I'm a dog owner myself. I think the saddest thing about dogs is their short lifetimes. You get to see them live and die. Imagine if you could just rejuvenate them.
Yeah.
So, yes, of course.
Just like thinking, say you're thinking markets. We're a bird family, and they live there really long as well. But I can imagine
yes,
plasma exchange into one. But no, this is it's very exciting stuff, Harold. We've watched you progress with this, and I watched.
Here it is. We're a relatively small company.
Mm-hmm.
Really, the workers are me and Jay.
Mm-hmm.
I'm 77 years old. Jay is young, hardy, hardworking, very strong, smart, self-made millionaire with a degree in finance. So this should be as big as the Manhattan Project because we're not trying to kill millions of people. We're trying to save millions of people. But that's the way it is now. And
isn't that how Genentech started?
Yeah, we will succeed anyway. That's the truth. We're already succeeding. So
no doubt you're on the right path here.
Yeah.
But so for everybody that's going to be listening to this episode on the podcast or watching on our YouTube channel, you've been listening to Dr. Harold Katcher today, Chief Scientific Officer at Yuvan Research. September 4th, his book, The Illusion of Knowledge, will be coming out, so pick up a copy. Harold, I want to take time to say thank you for taking the time out of your schedule to do this. Thanks for what you're up to. And as we say on our show, thanks for helping to create the better tomorrow through these programs. I really appreciate your time today.
My pleasure.
Good seeing
my obligation. You. Good seeing you. Nice meeting you, Ira.
Nice meeting you too.